Watch this enzyme settle onto a double helix and score one strand. The other strand holds. No break, no loose ends. And the gene it is about to write in is already here, clipped onto the enzyme itself, hanging a few nanometers from the scratch. That gene is more than 2,000 letters long, and it goes into the chromosome without the chromosome ever coming apart. Every CRISPR therapy in use today does the opposite. It severs both strands of a chromosome and hands the cell a template, hoping the repair machinery copies it in. Usually, that works. Sometimes the cell chews the loose ends instead and leaves a scar where the gene was meant to go. And when two chromosomes are broken at once, the cell can join the wrong ends together, a translocation, the kind of rearrangement you screen a cancer patient's cells for. In this paper's T-cell experiments, cutting produced translocations in about 1.8 of every 1,000 cells. The nick produced none the authors could detect. The work comes from Yanmin Gao and colleagues in Haifeng Wang's lab at Tsinghua University, published in Nature on the 22nd of July. They call it KNIT editing, kilobase scale nickase targeting. And the obvious question is why nobody did this earlier. A gentler injury? Sounds like it should have been the first thing anyone tried. The answer is that a nick is too small to get the cell's attention. Break both strands, and the cell treats it as an emergency. It stops, calls in repair machinery, and in that scramble, a donor template you supplied can get copied in. Nick one strand, and the cell barely reacts. It still has the opposite strand right there as a perfect reference, so it reseals the gap and moves on without ever consulting your template. Nicks were known to support small, precise edits. Nobody had made one insert a gene. So the team went looking for a way to make a nick productive, and they tried the things you would try. They fused repair proteins onto the nickase, including Rad51, whose job is to go find a matching template. And that helped, roughly twofold. They shut down the competing repair pathway to push the cell toward using the template. That did nothing. The move that worked was not about repair at all. They fused the nickase to a protein called monomeric streptavidin and tagged the donor DNA with biotin. Streptavidin and biotin grip each other about as tightly as two molecules can without actually forming a chemical bond. So the donor is no longer somewhere in the nucleus hoping to find the right address. It is clipped to the enzyme that is holding the address. That beat the bare nickase by 5.2 times, and the Rad51 version by 2.4. Proximity did the job that damage used to do. Underneath, the cell is still running its ordinary template-guided repair. Strip the matching flanking sequence off the donor, and insertion collapses. Take Rad51 or BRCA2 out of the cell, and it collapses. Same pathway gene editing has always depended on, reached through a much smaller wound. The evidence that it stayed gentle is in the alleles that did not get the gene. After a Cas9 cut, those alleles are littered with small insertions and deletions, the collateral of the break. After NIT, the same measurement came back at 0.46%, about 200 times lower, indistinguishable from a plain nick. Which has a strange practical consequence. The target site survives, so you can come back and edit the same spot again with the same guide. After Cas9, you often cannot, because the indels have eaten the sequence the guide was reading. Then, they made it greedier. In the second version of the editor, the nickase carries a tail of 24 repeating peptide tags. Each tag can catch its own streptavidin adapter. Each adapter carries its own copy of the gene. One enzyme sitting on one nick, reeling in a crowd of donor copies. At a safe-harbour site called AAVS1, that took insertion of a reporter gene from 56% of cells to 82%. Now, the cost, because there is one. A gentler wound is also a weaker invitation. at a gene called RAB11A, cutting put the payload into about 48% of cells. Nicking managed 30. Cleaner, but less of it. Size costs too. The platform carried inserts past 10 kilobases, big enough for most genes you would want to add, but that 10 kilobase insert landed in roughly a quarter of cells, against 80% for a small reporter. How well this works depends on which address you pick and how much DNA you are moving. There is one place where that trade is obviously worth taking, and it is the reason this paper is interesting rather than merely elegant. A CAR T-cell is a patient's own T-cell, given a new receptor that lets it recognize their cancer. The standard way to install that receptor is a lentivirus, which carries the gene in and drops it into the genome wherever it happens to land. It works. These therapies have put people with otherwise fatal leukemias into lasting remission. But you do not choose the address, and if the gene lands next to something that controls how a cell grows, it can turn that control up. In 2024, after reports of T-cell cancers appearing in treated patients, the FDA required a boxed warning across every approved CD19 and BCMA-directed CAR T product. Not a recall. These therapies are still the right treatment for a lot of people, but it is a class-wide label change and lifelong monitoring on a product that already costs several $100,000 for a single dose, and the virus is a large part of why it costs that. KNIT puts the same receptor at an address you choose, with no virus and no break. In primary human T cells, the team inserted a 2,394 base pair anti-CD19 receptor into the ACTB gene, and a slightly larger version into TRAC, the T-cell receptor gene, where knocking out the natural receptor is itself useful. The second-generation editor reached about a third of the cells, up to 40%. A CAR T trial that already worked in patients ran at roughly 20. The viability difference is the striking part. 8 days after editing, the KNIT cultures had 10 times as many living cells as the Cas9 ones. Cutting a T-cell's chromosomes is hard on the T-cell. Those KNIT-made cells killed CD19-positive leukemia cells and slowed tumor growth in mice. The other application answers a different problem. Lysosomal acid lipase deficiency comes from mutations in a gene called LIPA, and there are a great many ways to break it. Older clinical summaries list around 60 disease-causing mutations. Current variant databases list hundreds. If your tool repairs single letters, you need a different edit for almost every family. The team built cells carrying one version, a single letter change that does not alter the protein sequence at all, but sits right at a splice site. So the cell skips an entire exon when it reads the gene, and the protein comes out useless. Instead of fixing that letter, they inserted a whole working copy of LIPA. At a safe-harbour site, expression came back above normal. At the gene's own address, about 39% of wild type. One construct and which mutation you happen to carry stops mattering. Now, the limits. Everything in this paper is cultured human cells, mouse fibroblasts, and one mouse tumor model. Nobody has been treated. The repair pathway KNIT depends on runs mainly in cells that are dividing, so a liver cell sitting quietly in a body is a much harder target, which is why the realistic near-term use is outside the body. Take the cells out, edit them, put them back. The donor DNA is currently made by PCR and tagged with biotin, and the authors themselves flag that as a likely bottleneck at manufacturing scale. And the translocations are reduced as far as their assay can see, which is not the same as absent. What actually changed here is an assumption. Cutting was never the goal. It was the price of getting the cell's attention, and everyone paid it because there was no other way to be noticed. This paper buys that attention differently by holding the gene against the exact spot where the repair is about to happen, and most of what made the cut expensive goes away with it. The efficiency is not free, And at some addresses, the old cut still wins. But for the one place gene insertion is already a product being sold to patients, a version that leaves the chromosome in one piece is worth doing the arithmetic on.